Good morning, students. Today we're going to talk about the introduction and history of gene. So we have to know what is gene. Gene is a specific sequence of DNA. So we have adenine, guanine, thymine, cytosine. So these are the base pairs which will be present in a DNA. So this is what the gene. The whole contains genetic informations required for making a specific proteins. So DNA will be converted to RNA, RNA will be converted to protein. So that is what the basic things we have to going to talk about. So we have two types of genes, prokaryotic genes and eukaryotic genes. So which is normally seen in prokaryote is a basic gene which is present in bacteria and cyanobacteria and all types of bacteria. Coming for the eukaryotic, that is nothing but higher organisms. So lower organism is prokaryotes and eukaryotes is higher organisms such as plant and animal. So we all come under the eukaryotic genes. And based on the activity, we can talk about housekeeping genes, which are always active. And specific genes, they will be only active, only particular conditions, and the rest of the time they will be not working. So only specific time they will be working. So coming for the other base types of genes, based on behavior, we can talk about basic genes, the fundamental genes, lethal genes, that is only for the death process. Coming for the multiple genes, when two or more pairs of independent genes act together to produce a single phenotypic trait. So we have multiple genes, which is maximum more than one or two, it is working out to produce a single work. Cumulative genes, some genes have additive effects on the action of other genes. So cumulative, which is having one other action of the other things. And pleiotropic genes, which will be having the changes more than one character which is being brought out. And modifying gene, the gene which cannot produce a character by itself but interact with other to produce a modified effect. So they will not have their own things to do, but they will get interact with other things and they will be completing the work. So that genes will be known as modifying genes. Inhibitory genes, the genes which suppress or inhibit the expression of other gene is called inhibitory gene. So now we will move on to the basic of genes. So what is the term genes, what is genetics, we have to know. So all these are coming from the George Mendel. It was in the year 1865. He was only talking about the experiments based on with the peas, and he was talking about what is gene, what is inherited factors, everything. So this is a concept where you can see a cell which is having a nucleus. Inside the nucleus we can talk about the chromosome. The chromosome is nothing but a network of genes, so it is finally having your functional proteins. So this is what the things happen. So coming for the prokaryotic gene structure, you will be having a promoter starting and a terminator ending. In between you have the coding sequences, which is RNA coding sequences. So initiation and termination is very important for the processing. And coming for the regions, that is promoter, coding sequence and terminator, these three are the very important for the processings. And the regions 5' and 3' will be the upstream and downstream. So this is what the region where you have -35, -10, and where the RNA will be starting their transcription, and finally we go for the protein productions. So this is what the process we will be doing at Tata box TATAT, and you have the -10 sequences, -10 degree and -35, they will be going on, and they will be recognizing these factors. So this coding sequences, RNA coding sequences, they have the processing which will be copied as RNAs. That is what they code. So they will be ready to occupy as a RNA. So they have the starting and ending. Starting you have the initiator codon, and you have starting you have the initiator, and it will go with the along with the elongation process, and finally you have the termination codons, and you have extrons and introns. So introns are uninterrupted, so they are not needed for coding, so only exon we need. So we'll have this upstream elements, downstream elements, everything. So these are the basic things which will help for the prokaryote. Coming for the eukaryotes, comparatively eukaryotes will be more complex, and they're having exons, introns, promoter sequences, terminator sequences, upstream sequences, downstream sequences, enhancers, silencers, and signals, everything. So this is a picture which will explain you about the process. You have the regulation sequence which will guide you the process, promoter sequences where to store, and you have the 5 dash 3 dash translation units. You have the introns and exons. So the exons are coding sequences, introns are non-coding sequences, so we have to remove. So coming in detail, so exons is coding sequences which will be transcribed, and then they go for the translation. Then they will be coding for the amino acids. They will be varying in numbers, specific length, and they will be starting with the 5 dash to 3 dash ends. They will be having the UTR region where it is untranslated. So coming for the introns, you have the coding sequence which is separated by non-coding sequences called introns. So they are non-coding sequences. They are removed primarily when they are coming for the mature RNA. All introns had the base sequences with the GT and 5' and in AG in 3'. Introns were first discovered in the year 1987, and they found it about their sequences. So what is the role of these introns? They do not specify the protein productions but are important for cellular activities. They also encodes RNAs at specific gene expression level, and they also have the controlling capacity of transcription and mRNA processing, and they also help for different combinational alternative splicing mechanisms. Also it is important, an important role in evolution by facilitating recombinations between exons. So we have the promoters, we have 5' end upstream, and we have 3' end downstream. So in promoter we should have the core promoter regions, which is about 34 degree minus 34 degree site, where that is binding the factors, transcriptional factors, and promotional promoter which will have in the basic regulatory elements which will guiding the processing. RNA polymerase 2 will help for the transcription. Coming for the 5' N, you have the upstream, and 3' N, you have the downstream. So this is several functions including mRNA transport and initiation of translation and signal for addition of cap. So 7-methyl guanosine is added, 5' N mRNA, and stabilization of mRNA. Downstream you have to add mRNA, and stability will be attached with a poly A tail, and you have the termination part there, that is a codon TAA, and you have the sequences like poly A tails as AATTA. You also have terminator which has been recognized by the enzymes or polymerase, and they will stop the transcription. Enhancer will have the gene which is responsible for the production of few thousands of the genes for transcribing. Silencers will put down the expressions of the gene when it is needed. So coming for the what are the features of the gene. So what is normal gene can in each organism is having the chromosomes part. So they are very important for the chromosomes location. So only the gene will help you for the locations. The genes are arranged in a single linear order like beads on a string. So they are arranged as a beads as a string. Each gene will have their own specific positions, will have locus, and if the position of gene changes, then the whole character will be changed. So genes can be transmitted from parent to the offspring. So it is being transmitted, so it can be traveled from one generation to other generation. Genes may exist in several alternate formed, which is known as alleles, and also genes capable of combined together and can be replicated during a cell division. So it has been increasing the number of genes based on the replications, and they undergo several changes in their position and composition, which will result in your mutations. So mutation will happen only when there is a dramatic changes in their position or their structure or some changes in their genes. And also genes are capable of self duplication, providing their own exact copies. So genes make their own copies. So these are the salient features of genes. So basic genes, genetics, alleles, locus, every terms is very important to understand about the molecular biology and genetics. So this is about the gene, and thank you.